Good day everyone, my name is Dante Panlang Jr. and we're going to tackle about the abstract of the thesis entitled A dsRNA or double-stranded RNA-based screen identifies novel proteins involved in Drosophila Hh signaling pathway by Maggie's led in Holgram Laboratory. The Hh signaling pathway is known for the patterning of organs during organogenesis or formation of organs after embryogenesis or formation of embryo during development. In mammals like us, the aberrant type of Hh signaling pathway is responsible for formation of cancers. Thus, a further understanding of this Hh signaling pathway could lead to other treatments, since there are still many studies about Hh signaling pathway that remain unknown. The Hh signaling pathway in Drosophila melanogaster, a common fruit fly, is responsible for the patterning of the wing. So they use the UAS-GL4 screen and MS1096 GL4 driver to knock down the gene expression in the wing. It was performed in a sensitized fused 1 mutant background to indicate the presence or absence of FU1 phenotype in Drosophila. It was possible to determine whether loss of a particular gene had a significant effect on Hh signaling by observing the wing pattern. 11% of the Drosophila genome has been covered by the screen. 29 genes out of 1,590 were strong enhancers, and 11 of them were suppressors of FU1 phenotype. Megator, or MTOR, is a gene that strongly enhances FU1 phenotype. Thus, a knockdown of MTOR with RNA interference or RNAi decreased decapentaplegic and Collier HH target genes. The involvement of MTOR gene in the Hh signaling pathway will be discussed in the research.
So, good day, Ms. Yoko. I'm Ryan Tristan S. Gahudo, and we're going to present a sample thesis of a biology student. So, the thesis is entitled A dsRNA Based Screen Identifies Novel Proteins Involved in Drosophila Hedgehog Signaling Pathway, written by Maggie's led. So, I will going to discuss the introduction. So, first, what is introduction? Introduction falls under the chapter 1 of your thesis. So, at this part, you're going to give a teaser about your study or your research. So, in addition to that, you're going to state here the general topic and give some background or overview about your study. And also, introduction is the beginning of your paper. So, the main goal of this part is to catch the attention of your reader. So, on our sample thesis, the introduction, here, the author's introduction give an overview about how life begins from a single cell and the signal transduction pathway, specifically about the hedgehog or HH signaling pathway. The author gives some overview about the role of HH in development of the different organs, forming of wings, and nervous system of Drosophila or the common fruit fly. So at this, the overview also stated the capability of it to cure cancer. So, it is stated by the author in the abstract of the thesis. So that the furthest understanding of HH could lead to a new treatment. So, the next part is the RRL or the review of related literature, or it is known as literature survey. So, this portion falls under chapter 2 of your thesis naman. So, at this part, it contains the different data and information from different references such as books, magazines, journals, and etc. So, also, it is an extensive and up-to-date assessment of previous research regarding your topic. So, at this chapter, it is divided into two parts, the local and the foreign study. When we say local study, it is conducted in your country, while the foreign study naman is conducted outside your country or in other countries. So, in writing review of related literature, there are some few things that we should remember. So, first is, we must know how to cite or quote a statement properly. Hindi basta-basta kukuha ka na lang, then put it in your thesis. So, there are proper way of citing it. So, next is, it should be organized to cover the specific problem. Then, last, the unpublished material should not be older than 5 years if possible. And in addition to that, you must take note of the entire evidences gathered by the previous researcher. So it can, and lastly, on the last part of your thesis, you must have a statement how this old study helped your current study and relate it to your problem. So, on our sample thesis naman, the researcher state the different study about the HH gene. So the researcher presented it into the 4 parts. So the first is the studying HH signaling in Drosophila or the common fruit fly. Next is an screening for novel pathway components, and last but not the least is the importance of HH signaling. So these four studies states the different studies conducted to find out the significance of HH gene in organisms.
These are the materials and methods that are used in the research. Drosophila toxin culture that contains RNAi or RNA interference from Vienna Drosophila RNA Center, Drosophila Genetic Research Center, Kyoto Stock Center, and Bloomington Stock Center. The experimental fly crosses are obtained using 4 processes. In fused 1 screen, females with FU1 mutation and MS1096GL4 in their X chromosome were crossed to males with RNAi or RNA interference. The offspring will be able to suppress or enhance the FU1 phenotype when screened. In imaginal disc staining, RNAi or RNA interference containing flies were crossed to flies with WFU1, APGL, CYO, YW, DPP, LAC2, APGAL4, CYO, Y positive or W, DPP, LAC2, APGL4, CYO, UAS, GPP, TB. The results will be discussed later on. In generation of somatic clones, mutant cell clones were generated to FLP-mediated recombination. While in HH-MRT interaction, adult wings are being imaged and the ratio of the area between veins 3 and 4 to the total area of the wing was determined by image-based software.
Good day, Ma'am Yokomia Mendoza. My name is Alda Sofia Sibaro, and I am going to report about the results section of a thesis. What does a results section of a thesis have? Well, results section of the thesis have the applied methodology to know the study has reached its objective. The results section is particularly necessary if your paper includes data generated from your own research. Results have 3 rules. Number 1, the results should be based upon the methodology. The results section of the research paper is where you report the findings of your study based upon the methodology you applied to gather information. Number 2 rule. The results should be in logical sequence unfavorable on something. The results section should simply state the findings of the research arranged in the logical sequence without bias or interpretation. Number 3. The results should be written in past tense. Now, for the example we got from internet, the result of the thesis we get The thesis we get titled A dsRNA-based screen identifies novel proteins involved in Drosophila hedgehog signaling pathway. Letter A. Genome-wide screening. A screen was performed by crossing flies containing RNA against no target genes to a fly stock with the driver and background sensitized by a hypomorphic mutation. The mutation results in a characteristic vein phenotype, the fusion of the distal portions of wing veins 3 and 4. The progeny resulting from this process were scored for changes in the phenotype and classified as strong, weak enhancers or suppressors, depending upon the nature of the observed effect. 250 genes were screened as part of this project are likely to be involved in the regulation of the HH signaling pathway due to their enzymatic activity. These 250 genes were part of a larger effort to screen the entire Drosophila genome in order to identify novel proteins involved in the HH signaling pathway. And that is all my report.
Good day, Ma'am Yoko. My name is Erica Andre Arteriano, and I will discuss about Chapter 4. Chapter 4 are research findings and discussions. This chapter presents the results of the analysis. Let us know the Chapter 4 of our sample thesis. Chapter 4, letter B, immunohistochemical assays. What is the meaning of immunohistochemical assays? Immunohistochemical assays refers to the process of detecting antigens. Example given is protein in cells of a tissue sections by exploiting the principle of antibodies binding specifically to antigens in biological tissues. In this study, the expression of target genes was examined using an immunohistochemical approach in Drosophila larval imaginary. RNA lines were crossed to plants containing apterous therefore, which drove expression of the RNA in only the dorsal compartment of the radius. Changes in gene expression were determined by comparing the dorsal and ventral halves of the disc. Initially, only DPP, PTC, and Psi expression were analyzed for the RNA lines that were found to have an effect on the FU prime phenotype.
Hello ma'am, I'm Hansar Del Garcia. I will be reporting the Part C in results. Megator. Let's see. When we say megator is a protein that regulates GSC and GSC maintenance and what do you call this? The asymmetric division through the spindle assembly checkpoint. Let's see. In the first test, megator and PTC levels are injected in the wings of Drosophila. So as a result, in the knockdown of megator resulted a decrease in the target gene expression, and in the PTC or the PTC protein levels has no effect, but they saw a decrease in PTC transcript by using LACZ reporter. As a second test for MTOR was involved in general transcript. MTOR RNAi was crossed to flies that contains a ubiquitously expressed GFF, which means no difference at all. So the tests are done to see if there would be any changes in the wings, if some proteins or protein levels is injected in it. Let me see. I have here some keywords. Maybe you're wondering what is the meaning of HH. HH means hereditary hemochromatosis. It's also called genetic hemo. It's a genetically determined disorder in which mutation of certain genes involved in metabolism. Let's see. The whole thesis. No, not the whole thesis. The tests, rather. The test performed. Let's say no. The whole thesis study about the proteins, the RNAi, rather, if injected in Drosophila, what would be the result, and some protein levels like PTC. So, as I've said before, MTC, let's say, how do I say this? If I were to explain this by using DNA, a DNA strand, for example, a DNA strand is what do you call this? If a DNA, this, for example, this is a DNA strand, and they remove this to change to what do you call this? To replace it with the mediator or the RNAi that of course that will cause certain changes because the protein, for example, we have this DNA. In this DNA, the other strand is removed and the RNAi is inserted. Then, what is a function of protein? Protein produces enzymes that gives command to our body or body activity, let's say. And yeah, that's all. So the thesis just taught us my part just taught us what happened in the wings when they use mediator as a what do you call this, as a test sample for Drosophila's wing. So this is the end of my report. Sorry for some errors, I guess, because I'm not familiar with this topic, and besides, we're in basic biology and this topic is so complicated, so I can't expound it well. Thank you for your understanding, and for listening. Thank you.
Good day, Miss Yokomia Mendoza. I am Jasmine Faye P. Guadalupe. Today, my topic is about the acknowledgement, discussion, and the curriculum vitae of a research paper. Discussion. Discussion is the framing section, like introduction, which returns to the significance argument set up in your introduction. In this section, you will discuss how the hypothesis has been demonstrated by the new research and show how the field's knowledge has been changed by the addition of this new data. In this section, you must avoid rambling, guessing, or making logical leap beyond on what reasonably supported to your data. Discussion is set up as a dialogue of your result and the other theories. In other words, on every experimental result you want to talk about, to find other results from other publication bearing the relationship to your result that make your reader understand your research. Acknowledgement. Acknowledgement is the appropriate way to give credit to all individuals who have been helped during this project. For instances, by supporting financially and giving feedback on the text during its composition and revising stages. Generally, acknowledgement contains the full name of all individuals who are being thanked and a brief statement to what kind of help that the author received from each individual or group. Curriculum vitae. Curriculum vitae is the academic version of your resume. It is generally used when applying for an academic position, grants, research, and admission for some graduate programs. CV is a summary of your educational and professional backgrounds. It includes the full list of your publication, research, honors, and awards. The basic elements of curriculum vitae are 1. Personal information. 2. Educational areas of specialization and dissertation. 3. Awards, honors, and grants. 4. Experience and publications. 5. Professional activities and memberships. And 6. The reference. Curriculum vitae must be well organized and easy to read. Choose an effective format and be consistent and strategically place the most important information near the top and the left side of the pages.
Good day, Ma'am Yoko. My name is Erica Andre Arcarinion. I will discuss about Chapter 7 or Reference Chapter. Chapter 7 or Reference Chapter includes all the reference used in the study. This will prevent us from plagiarism, and it will prove that we did the research. Note, this is written alphabetically.